Hello, everyone. My name is Tegan Clary. I'm the Vice President of Marketing at Unchained Labs. I'll be your moderator today, and thanks for joining us. This will be a really exciting topic. We just recently launched some new capabilities on our Big Tuna instrument, and I think you're really going to enjoy the presentation today. We will have a Q&A session at the end of the presentation. To ask questions, all you have to do is find the little Q&A button in your Zoom navigation bar. Type in your question and we'll get to as many of them today as we can. And now I'd like to introduce Joe Barco, our Senior Director of Marketing at Unchained Labs. Today, Joe will walk us through how Big Tuna can prep gene therapy and vaccine samples easily and efficiently. Hey, Joe, happy Thursday.
Thanks, Tegan. Happy Thursday.
Nice to see you. And now I'll go ahead and hand it over to Joe. And Joe, go ahead and take it away. Looking forward to it.
Okay, thanks, Tegan. And thank you, everyone, for joining us this morning. So what I'm going to talk about is some ways that you may be able to offload some of your buffer exchange and sample prep work, especially for AAVs and LNPs, to Big Tuna. I'm sure many of you think about buffer exchange the way I do, which is it's a chore, and it's just something you have to do to get on to doing something that you want to do that's more important. Personally, my least favorite chore is folding clothes, and I will push this off as long as possible, even pushing off the task before, which is washing the clothes, in order to avoid folding them. Now, if you do a lot of sample work for biologics, you know that you don't really have a whole lot of flexibility in terms of how much you can push off the task, because the whole point of buffer exchange is to get your biologics into a buffer where it's going to be happy and stable. And I think nowhere is this more apparent than in the case of lipid nanoparticles. So LNPs are formed in the presence of a buffer where it cannot stay for very long because of an organic solvent being present. And so the whole point of buffer exchange is to make the molecule more stable. Other examples like viral vectors, AAVs, are produced in very dilute concentrations, oftentimes orders of magnitude below where it needs to be dosable, or even from a practical handling standpoint. And they need to be concentrated to do any further characterization work on it. Things like nucleic acids and proteins and antibodies have been doing these for a long time. And so the issue for them is more of a throughput issue. They oftentimes end up in a solution where it's coming off of a resin or a column, and it may be dilute. It may also be in a buffer which has too much salt or the wrong pH or just the wrong buffer period. And it needs to be exchanged into a buffer where it can be conjugated or stored or characterized for stability. Especially in the antibody side, you have a lot of work that goes into formulation, optimization, and screening, which for things like new lipid nanoparticles and AAVs, which are fairly new from a biologics and storage standpoint, are eventually coming. But you'll need some way to do some characterization of formulation to ensure that there is a long and stable shelf life to that molecule. Because buffer exchange is ubiquitous, there's many ways to do it. And most of these methods are low tech. And dialysis is a good example of this, where the best thing about dialysis is you don't have to be there to do it. You can start it, leave for a day, and then come back and take your sample. The trade-off for being fully walk away is that it doesn't end up at the concentration you want. It almost always is more dilute than when you started. And so you have to then do a separate concentration step to it. Centrifugation filters are pretty much the opposite problem. You can do them fast, you can do them at the volume you want, but you can't walk away from it. And you're stuck at the centrifuge going back and forth with just not enough time to kind of do anything else, but too much time that you're not really spending it in any productive manner. And if you have to do multiple samples, forget it, because you have to now worry about, you always have one or two that just seem to run slower or different than the rest. And tracking that while you're also tracking samples that are running fine is just a hassle. TFF is the most high tech of these three methods. The trade-off for that is that you can't just walk up and use it. You have to set up the pumps, have to set up the cassette. The cassette is expensive. And you also have to worry about the volume being the right volume. So you can't run multiple sample types at once. It's much more of a larger scale thing. So normally 50 mils and above to start with. When we developed Big Tuna, we were looking for a solution that could allow you to do buffer exchange in a robust manner. Big Tuna is a fully automated buffer exchange and concentration platform that can handle samples in many different formats. You can work with as little as 100 microliters a sample in a 96-well format, or nearly 50 mils in a 24 mil format. or 24 well format. What Big Tuna will do, what we'll talk about today, is specifically how it can work with some of the molecules shown on the left-hand side. And as Tegan mentioned earlier, things that are new features and capabilities that have just been launched on the platform. First, let me talk a little bit about Big Tuna to familiarize yourself with the data I'm going to show in a moment. Big Tuna is a filtration plate-based exchange. We use a consumable called the Unfilter, which we've developed ourselves at Unchained Labs, and we pressure test 100% of the wells before the plates go out the door. The 96 well format is an SBS format. The working range of that is 100 microliters to 450 microliters per sample or per well. And then the 24 well format has a working range of 450 microliters to 8 mils per well. Both have regenerated cellulose membranes at varying molecular weight cutoffs of 10, 30, and 100 kilodaltons. From a cost standpoint, this is comparable to a centrifugal filter, except in a format that is much more amenable to handling many samples at once. The way Big Tuna works is it uses a modified ultrafiltration diafiltration process. So you take your samples, put them in the Unfilter. That goes into a buffer exchange chamber. It measures the volume with an acoustic non-contact sensor, and then starts a gentle pressurization cycle with orbital mixing. After some period of time of pressurization, it stops, measures the volume again with the sensor, and determines how much buffer has been removed from the sample. It then goes back and adds refills with either the new buffer or the sample itself, and starts the process again. So it repeats that cycle over and over until either the desired volume has been added to the sample, or it's been exchanged into the new buffer. This filtration process is adaptive, meaning that it will do a modest pressurization at the outset, determine the flow rate for the samples inside, and then set future pressurization times based on how the flow rate is working. So what that does is it ensures that you are finishing the process as efficiently as possible. Each well is individually tracked, because as I mentioned before, sometimes samples will run differently for changing the sample concentration or the buffer, or for who knows what reason. So what we want to do is make sure that in the process of running it, that those fast samples aren't drying out while you're waiting for the slow samples to come along. And so that individual volume tracking ensures that every sample that's being run is being run in the best possible manner. There is a programmable pressure level, and what that does is it ensures that your sample is being handled as robustly as possible. Most samples are exchanged and concentrated at 60 psi, or about 4 bar, but we found that certain sample types like AAVs and low concentrations of nucleic acids or proteins can actually be exchanged and concentrated in as little as 15 psi, or about 1 bar. Now we orbital mix as part of the process to make this as efficient as possible. Now I'm going to contrast that with dead-end filtration, like a centrifugal filter shown on the left-hand side. In dead-end ultrafiltration, you're concentrating your protein at the membrane surface as you're exchanging. That process slows down the flow rate and also creates a concentration gradient within the sample. That could have a couple of untoward effects, things like clogging and fouling the membrane. It could potentially aggregate because your sample is at a higher concentration at the membrane than it would be otherwise. And we've seen many times in even high concentration proteins that it will gel on the surface. So to avoid these problems, Big Tuna does an orbital mixing process. That orbital mixing process ensures that the sample is staying uniform in the well, and that flow is consistent and uniform during the exchange process. So not only does that make the exchange go faster, it also ensures that your sample is being handled in the best possible way. By automating this process, what we do is we give a degree of process control that you would not have on a manual method. So first, you have the percent removal, which is how much buffer gets removed in each pressurization cycle. For things that are very dilute or very robust, you can do a high percent removal, which will help you go through the process of exchanging and concentrating faster. For samples that maybe you're not as familiar with, or they're high concentration, or you're concerned about the buffer change being too drastic, you can do a lower percent removal. And that ensures that your sample is gradually introduced to the new buffer without putting your protein or sample under further stress. The percent exchange is the total amount of buffer replaced. Most times when you're doing buffer exchange, you want complete exchange percent, something like 96 or 99% above or above. For things where you're trying to establish sort of a flow property or understand, characterize some earlier sample types, you may want to just do a lower percent exchange just to get a sense about how things flow. And then finally, you have the concentration step. The concentration, you can concentrate directly without doing buffer exchange. You can do that with the full range of the plate. So for a 24-well plate, you can go from 8 mils to 450 microliters. There's an application on the system where you can actually continue to add sample back in to that 24-well plate, and you can do as much over 100-fold concentration of that sample. Now, just looking at these molecules, you can probably guess that it can't do everything exactly the same. There are very different sizes. They get presented in buffers. They give it much different flow properties. And so what we've done on Big Tuna is we've created presets based on the molecule type and the concentration. And what that does is it allows someone who's never used the machine or never walked up and used Big Tuna to be able to kind of navigate their way through and establish how they should run a sample. Because what we've done is we've already figured out the best way that we think that sample is going to run. So even though that pressure cycle is adaptive, knowing that it should probably start at a lower pressure, or we should work with it at a different percent removal, is a great starting point for someone who's never used the machine before. Now, over time, you'll become an expert in Big Tuna, which won't take that much time at all. And then at that point, what you can do is you can always override those presets and run it in the way that makes the best sense for your sample. I'm going to spend the rest of the talk talking about some proof of concept examples for these gene therapy vectors. So let me start with lipid nanoparticles. So LNPs are formed by the combination of two different phases. You have an aqueous phase, which contains your genetic payload and a surfactant. And you have an organic phase, which has an organic solvent, and then other polymers that would make up the LNP. You combine these two phases and create an emulsion. And within that emulsion, the nanoparticle is formed spontaneously, ideally encapsulating the genetic payload. Now, the problem is that you need to immediately remove that ethanol, which is usually the organic phase, and to stabilize the molecule with the payload intact. And so as soon as that emulsion is formed, you have to exchange it. And so the problem we're trying to address here is to quickly remove the organic solvent and to also concentrate the sample, which is now, by the process of making it, more dilute than desirable. For this proof of concept work, what we did is we took this firefly luciferase mRNA encapsulated LNP, and we had this in 10% ethanol. We concentrated it threefold to both exchange out the ethanol and put it directly into PBS and to make the molecule more stable. For this, we used a plate type 100 kilodalton unfiltered 24, so the larger volume, and then the higher molecular weight cutoff to ensure that flow was as fast as possible. We used an LNP preset that's new on the instrument to establish, to do the exchange, which is at 60 PSI in this particular case. This exchange and concentration was done in about 90 minutes. And so what we're confirming is a couple of things. One is that we can hit the concentration target, which is the sample concentration by about threefold, which we did achieve that concentration target. Big Tuna will measure the volume as it goes. And at the end of the process, we can establish that we've reduced the volume by a factor of 3. And for percent encapsulation, we used a ribo-green assay. And we determined the percent encapsulation was 98% before we started. And then when we were finished, we were within a couple percent of that target. So we did not damage the LNP in the process of exchanging out the ethanol. And we did this all in one step without the sample ever having to leave the chamber or without having to stop and do anything else to it. Next example is talking about the concentrations. So many times samples come more dilute. And what we're looking at, especially for AAVs in this case, is some way to concentrate samples down relatively quickly without a lot of user intervention required. And what we did was we created a new application to enable this on Big Tuna. Now, normally in unfiltered 24, we can go from 8 mils to 450 microliters. But what happens if you have more than 8 mils? So what we established is a way to do this where you add 8 mils to the unfiltered 24. You then take your rest of your sample and you can put it on reservoirs that would sit on the deck. So you can add an additional 40 mils of each sample, and you can do this for 24 samples simultaneously. So that unfilter is in the exchange chamber. It's pressurized just like the buffer exchange. However, unlike buffer exchange, after that pressurization and volume measurement step, you go back and add sample from a reservoir instead of the buffer. And so it will repeat that cycle until you've added the volume requested by the operator and end at a point where you have 8 mils of sample in each well in the unfiltered 24. So you can process 24 samples in parallel. And then if you want to concentrate further, you can still do that on that same unfilter, which is you just take that 8 mil plate and then you can concentrate it down to 450 microliters. So you have the 2 reduction steps will bring you to over 100-fold volume reduction in one plate in 2 steps. So for this, what we did was we took AAV9 at a volume of 18 mils, and we did do this in the 2 steps I mentioned. So you take the sample, reduce it at 8 mils to the unfiltered 24, have 10 mils in a reservoir on the deck. You reduce that 18 mils down to 8 mils, and then you run a concentrate only step. And in this case, we want to go from 8 mils to 500 microliters. And we're going to use the unfiltered 24 at 30 kilodalton molecular weight cutoff. That's a new plate, and also 30 kilodaltons is the right molecular weight cutoff for AAVs. And for both of these, we're going to use an AAV preset. And the preset runs at 15 psi. We are checking our concentration. So we're at about 5E11 to start with, and we want to get to about 1.7 e to the 13th as the 36-fold concentration. And so we're within 5% of that target. For the volume itself, we go from 18 mils to 500 microliters. And again, we're about 5% of that target. We're using a capsid empty full ratio test. There's an application on our Stunner platform called AAV Quant, which allows you to get a measurement on the empty full capsid ratio. So we start with about 2/3 of the capsid full, and we end with about 75% full. So from this whole process, we do a 36-fold concentration. We're done in about an hour and a half, and then the capsid payload is still intact within the sample. Now, since this is a gene therapy discussion, I'm going to talk a little bit about the nucleic acid desalting. This is possible on many platforms. The benefit of this on Big Tuna is because of the orbital mixing. It's uniform across the plate. Other methods by vacuum filtration oftentimes have edge effects or kind of the smile pattern of getting the right concentration. We don't have that on Big Tuna. So in this proof of concept experiment, we're going to take the double-stranded DNA that's in TE buffer, and we're going to concentrate that 3-fold. And we're going to do both of our plate types on this. So the 30 kilodalton plate, the unfiltered 96, and the unfiltered 24. So in this particular case, we're going to go from about 2 mg per mil to about 6 mg per mil. So we'll start with the unfiltered 96 data. We're a little bit above that target, but we're hitting the volume. We've confirmed on lunatic measurements that we have this done without any loss of material to the plate itself. And then in the unfiltered 24, same thing. Same concentrations, about 2 mg per mil to 6 mg per mil. We're going to go from 3 mils to 1 mil. We've established on Big Tuna that we have that volume. We've established on lunatic that we have the amount of DNA present, and we've hit the exchange targets on these. I hope what I've given you is a starting point to discuss how Big Tuna might be used for your particular sample types. So the best thing about Big Tuna is you can walk away from it. I'd say the next best thing is the software. And so we're happy to show you the software and demo that for you. But it's super easy to walk up and use it. And the benefit of super easy to walk up and use, and then the additional benefit of being able to walk away from it, you can do a lot of sample work in parallel for many different sample types. And finally, what I want to say is hopefully Big Tuna can take one of your chores off your hands, knowing that it's going to handle your sample at the appropriate cycle for your particular molecule and maintain with best robustness. Okay. Thank you so much for your time and attention, and I'm happy to answer any questions, and I'll turn it back over to Tegan to moderate questions. Thank you.
Awesome. Thanks, Joe, for that great overview of Big Tuna and the new capabilities. I'm excited that people are going to be able to use it for buffer exchange of these bigger complex molecules, cleaning up samples. And I think that concentration application is going to be really helpful for people. So thanks for taking us through that. We got a bunch of questions. And if anybody has any additional ones, please submit them. I'm looking at them right here, and I'm happy to answer pretty much anything you guys ask. So go ahead and send us in any questions, but let's go ahead and start talking through them. So Joe, I'm going to start with some questions that were more about the Big Tuna in general and how it works, and then we can get into some of the applications, okay? One of the first ones was, what kind of pressure are you using to perform the filtration with Big Tuna?
Yeah, so most of it is done at 60 psi, and that's generally high protein concentration, LNPs, things like and higher concentration nucleic acids. Those flow really relatively slow in comparison, and so they need that pressure, which is still not a whole lot of pressure when you compare to centrifugation. Things like AAVs and low concentration proteins run really fast, and so we actually reduce that down to 15 psi to ensure that that's flowing in a consistent manner.
Right. Okay. Another one, and I think, Joe, this will depend on the sample and the type, but just typically, how many buffer replacements or cycles typically happen on Big Tuna for prepping a sample or exchanging it from one buffer to another? Maybe I'll elaborate that to these AAV molecules or LNPs. What are we typically seeing?
Yeah, so we do those at generally a 33% removal. So if you're going to do this at if you're going to do a 96% exchange, that ends up, I'm doing the math off the top of my head, which is never a good idea, but I think it usually is 5 or 6 cycles. And those cycles on AAVs are really fast. So they're like 5 to 10 minutes per cycle. Things like protein concentrations, it's usually about the same number of cycles, but that pressurization time may be more. And I don't know, the unasked question perhaps is about the amount of buffer or the amount of tips to use. And so I'll just say a couple things on that. There's room on the deck for about 1 liter of buffer. And the way that the tips get used is you can actually reuse the same tips for buffer. So you don't go through more than a rack of tips. And if you're using the same buffer, you can go through like a dozen tips and that's it and be done.
Okay. Another one on the use of the Big Tuna. 2 questions. So do people need to use the unfiltered to do buffer exchange? Because it's something they have to use?
Yes.
Got it.
Yeah.
And then I know you mentioned I know you mentioned some of the application and you went into a couple, but what kind of molecular weight cutoff selections do we have available?
Yeah. So for both plate types, we have 10 kilodaltons, 30 kilodaltons, and 100 kilodaltons RC membrane. And then for the 96, we also have a 3 kilodalton option.
Okay, great. A couple of applications questions. I know you talked about a DNA cleanup experiment on there. Can it also handle mRNA samples? Do we have anybody that's doing that? We do.
Okay. we do, and it's a customer. I can't talk about their data. I mean, in our hands, we had for a proof of concept, we chose DNA, but we know from other customers' usage that mRNA works fine as well. Okay, great. And then recovery question on AAVs. You talked about the AAV example. What's the typical recovery look like for cleaning up and concentrating an AAV? It's 96 and above. 96% above is what we've seen in our hands. Okay. Another kind of processing question on the system. How do you handle samples with different flow rates? Like what do you do when that happens? Yeah, so what we do is we adjust the pressurization time. So every well gets measured, and as I said, there's a lot of times where some wells will just run faster than others. And what we do is we tailor the next pressurization time to the slow well. And so if the slow well is running, I should say, sorry, the fast well, which is the fast well, we don't want the fast well to dry out. And so we set the pressurization time to ensure that fast well not only not dries out, but hits that the percent removal target. And so what that may mean is that things that are running that you end up taking more cycles for that, but every sample is being handled in a way that you're going to get to the end of the experiment and they're all handled well, or they're all stable and good.
Okay. In your experience, Joe, can this type of buffer exchanger on Big Tuna handle AAV cleanup out of iodixonol?
Yes, I believe that's yes.
Yeah, I think we've worked with somebody on that before. So I think, yeah, the answer to that is yes, it's compatible with it, and I'm certain we're able to get it out of iodixonol. I will also give a plug for our stunner instrument for that, because we're also able to do a check on AAV molecules with stunner to see if iodixonol is still present or was present to begin with. So it's a nice tool to be able to clean these samples up out of iodixonol, and then also to be able to check that it's been removed. We see that in the UV-Vis spectrum on our stunner instrument. So if you want to talk to us about that specifically, reach out to us and we can talk to you about how to set up that kind of workflow. So, Joe, for LNP applications, there's a couple questions here. So what was the time for buffer exchange and the pressure used? And this is specific to some of our changes to Big Tuna, right? So first, maybe what's the pressure used? What's the setting for LNPs? And then typically how long did those kinds of experiments take?
Yeah, LNP, we use the default pressure, which is 60 PSI. So in terms of doing this development work, we just established that works fine. So the time is about 90 minutes overall. It'll probably be it's somewhat dependent on how many samples you're running at once, but it's about 90 minutes total. And we did check the size afterwards, and confirmed by DLS on stunner that we maintain the same size and PVI beforehand, before and after. And that's data from this experiment. I see who asked the question, so we can share that data if they're interested.
Sure. Yeah, and we can follow up on that specifically. There's another question here about the size of the particles after processing. And I think typically from what I've seen, I know what you've seen, we check it before and after. We don't see a lot of impact on size change on the molecule specifically. Of course, if you exchange it into something it doesn't like, that could happen. But typically, I think many people are getting out of ethanol or are concentrating into something that an AAV is happy in. So we typically don't see that because we're typically putting it in something that folks know they want it to be in. So yeah. Okay. Well, I think if the folks that want to talk to us a little bit more about the before and after measurements with a stunner and the processing of Big Tuna specifically for certain molecule types, just reach out to us and we'll talk to you more about that. Okay. A couple of other processing questions, Joe, about the instrument. Are all wells pressurized to the same pressure, or are they being handled individually?
They're all pressurized to the same pressure. So the chamber pressurizes the entire plate at once.
Okay. And then, this other question, how many wells are being run at a time for the runtime you've showed? And I don't know specifically which experiment somebody was asking, but maybe you can just give a general amount of runtime on the examples you shared.
Yeah, for AAVs and LNPs, it was 3 to 6 wells we did at once. For the DNA work, I don't think I had time shown on that. That was the full plate, so it was 96 or 24 wells at once.
Okay. And just to make sure I answer this one question here, have we checked any molecules for morphology or Subvis particles or anything post run, or is that something we haven't looked at yet?
We've looked at it by DLS, but we haven't looked at it specifically for particles. LNPs are a little are cloudier solutions, so it's a little harder to establish changes in that. But it's a good point. We can look at that, but we've only looked at it by DLS so far.
Okay. There's a few more quick ones here. Does Big Tuna have temperature control? We get asked that a lot.
Yes. It does not. So what we focused on, there's a couple things. One is that we focused on trying to get the exchange done as quickly as possible. And so most of these things are done within a few hours. The intent being that do it fast and get it into a condition that you need. I think the second thing is that we have in the past had another platform where we did exchange at 4 degrees was possible, and everyone the customers who used it didn't do it because it ran so incredibly slow. because viscosity is a function of temperature. And so it's going to run slower, and flow is a property of temperature. It's going to run slower at 4 degrees. So we focus more on getting it done at ambient and getting it done quickly. I think there's also a little bit of, I'll always say this when this question comes up, other methods, there's, I think, a little concern about stability that's maybe a little bit overwrought, which is that you have to, like, it doesn't have to be, if you're worried about injecting this into someone eventually, like, it should be stable at room temperature for a couple of hours. Like, it's just not, your stuff's much more robust than you think it is sometimes.
Yeah. I mean, we've had a lot of people using Big Tunas for a while now, and this really hasn't been an issue. But if you put it in something, again, it's not happy in, you're likely to find that out anyway. So, but yeah, I follow you.
We do a lot to mitigate the sample handling during the process. So it's in the exchange chamber. When the exchange is done, if you're not there to take the samples off, the chamber door stays closed, and that protects it from airflow in the room and keeps it at ambient temperature. So it's not exposed to the air.
Okay. All right. Well, this will be the last one. I don't see any others except this one. So do we use negative or positive pressure during filtration? Maybe you can just describe kind of how we pressurize the unfiltered plates, and then we'll wrap it up.
Yeah, so it's positive pressure. So plate is sitting on a stage that has orbital mixing. That whole stage is enclosed in the chamber. That whole chamber pressurizes at 15, 30, or 60 psi, depending on what sample preset you used.
Okay, great. All right. Well, hey, Joe, thanks for a great presentation. Thank all of you out there that asked questions. We appreciate that. I'm sure some of you have more questions. You can reach out to us at info@unchainedlabs.com, or reach out to somebody you know at Unchained Labs, Joe, myself, whoever. We're happy to talk to you more about Big Tuna, and specifically how it can work with these complex molecules and some of the new approaches that we've put into the system. We're really good at doing virtual demos. If you'd like to work with our team to actually see this thing, in a seminar first, and then work with us over Zoom to actually run some samples, just let us know. That's something we really love to do, and we love working with people that are interested in solving these kinds of problems. So thanks for your time today. Yeah, Joe, go ahead.
Sorry, we should do one more plug, which is we're doing a virtual demo in a couple of weeks.
Perfect. Yeah. Virtual demo in a couple of weeks, so you actually get to see the instrument live and see us running it and see how it works a little bit on the software. So yeah, please do join that. But if you can't wait and you want to talk to us before that, reach out and we will be happy to talk to you and work with you. So thanks again for your time today. And Joe, thanks again for a great presentation. Bye, everybody.
Bye.